Now we proceed to the second part of it, which is the photorespiration. And this is actually happens because, so by the way, Calvin cycle, the other name for it is C3 cycle. There's so many. Pentose phosphate cycle, C3 cycle, Calvin cycle, dark cycle, so many other names, synonyms. However, the star enzyme involved in Calvin cycle, the Rubisco, Ribulose, bisphosphate, carboxylase, oxygenase, is an ancient enzyme. Therefore, it has the issue not to be specific enough to catalyze single sole reaction, which is the carbon reaction up here, right? If it proceeds with this carboxylation reaction, then that's fine. You get your phosphoglycerate that can be reduced to become your sugar precursor. However, the issue with Rubisco is it can add up oxygen to the RUBP and proceed with, many like to call it a wasteful process. I would like to call it metabolically expensive process called photorespiration. When it proceeds this way, it will create two molecules. The first molecule is the regular phosphoglycerate to proceed with the regular Calvin cycle. The problem is here. It creates the two carbon product, not three carbon, two carbon product called 2-phosphoglycolate. This thing actually is a bit toxic to the plant. So what the plant does, the plant needs to eliminate this or need to turn this back so that I can reuse this carbon for the sake of making sugar precursor again. Because this thing, another problem is it cannot be converted directly from here, jump to here. Plants do not have the enzyme. Unless some kind of mutation happens, then we do not have this photorespiratory issue, right? So photorespiration is a type of respiration, meaning that it consumes ATP, it breaks down something to get something, but the trouble with photorespiration is it's a lengthy and expensive process. To start with, photorespiration involves three organelles. Chloroplast, from where it started originally, then peroxisome, then the third one is mitochondria. Okay, mitochondrion, that's the singular, mitochondria, that's the plural of it. So these three organelles, without the oxygenation reaction done by the Rubisco, these two really are of the question. Chloroplast alone is enough to make the sugar precursor. Now, since the plants have produced this toxic two-carbon product, phosphoglycolate, these need to be turned into something useful again. So the whole idea is from 2-phosphoglycolate, how to get back to 3-phosphoglycerate. You see, it's just from here to here, but there is no enzyme to do this. So in order to do that, plant has to go all this path, go back, go down to the peroxisome, mitochondrion, coming back to peroxisome, coming back to chloroplast. It's such a long process. So you are equipped now with the enlarged version of this so that you can study on later. Right. So for the second page of this is the summary of the reaction that's going to happen. But let's be realistic. Yeah?
Why there is a question mark?
Oh, I'll come to that in a bit. So you see these light purple boxes here. These are called transporters. Transporters meaning that it's like a portal or doors from which molecules can exit or enter. Okay. 2 years ago, when I taught this photorespiration, people have not confirmed to find the name of this transporter. So this transporter actually, I can give you the name. So it's PG GG1 and BAS. So P stands for plastidial glycolate glycerate, number one, and BAS, what is that? This is, by the way, this is transporter, okay? This is called transporter. This BASS6 is called symporter, meaning that it one door used by many people, the sim. BAS is stand for bile acid sodium symporter 6. Whenever you see this question mark, meaning that people have not found the name or the protein involved to make the. So this is the research opportunity actually to study this. I tell you, it's not easy. It's not easy. In my thesis, there is a technique that can be used to detect this. Okay, if you ever read my thesis in chapter 5, I describe there is a technique to detect this. Right. Okay. Coming back to this cycle. I just want to be realistic about this. The realistic bit that I'm talking about is you to cover this in one lecture. You need to find a happy, peaceful place to read it and to absorb. But what I can assist with you here is to create the story so that you can see that it starts with something, then it come back to something, right?